welcome to hematology. This entire lecture is going to be dedicated to just the red cell. If you're able to achieve the objectives, then it should be a good starting point for you, especially if you're trying to follow along the lectures. I also have these additional concept checks here. Again, if you're able to answer these, then you will be on a good path. So the red cell was one of the first cellular elements that was ever observed, and they play a vital role in being able to carry oxygen from the lungs to the tissues, as well as removing waste products from the tissues and allowing them to be transported to the lungs, kidney, and liver for removal. Excuse me. The shape of a normal RBC will be this biconcave disc. So biconcave just means that we have two sides that are caving in. And it's about 7 to 8 micrometers in diameter. They are by far the most abundant type of cellular element found in whole blood, where males have a count of about 4.5 to 5.5, and females having a slightly lower count, 4.0 to 5.0. Erythropoiesis, if you love your root words, erythro just meaning referring back to erythrocytes or red cells, and poiesis, which is the generation. So all we're talking about is just the process of making RBCs in the bone marrow. And the reason that we need to do this is help the body meet oxygen demands. Erythropoiesis is heavily regulated by hormones, especially erythropoietin, which is generated from the kidney. Red cells live for about 120 days, and they're cold and pitted by macrophages in the spleen. They can also be cold and pitted by the liver and the bone marrow if needed. Generally, any certain disease processes can cause either increased RBC destruction and or decreased RBC production. If you have a combination of both or one of these, then your body's balances will go out of whack. So there's 6 stages that we talk about in erythropoiesis. So generally, as the RBC matures, we're going to see that the size starts shrinking down, and this nucleus also relatively gets smaller, especially compared to its cytoplasm. The size of the nucleus compared to the size of the cytoplasm is called the NC ratio. And what happens is that nucleus becomes so condensed that it'll eventually become extruded from the cell or completely removed. And last but not least, we're going to go from this blue cytoplasm to this pink and salmon color. But that's the general traits that we see as the red cell develops. So the stages go as follows. We go from the hematopoietic stem cell to the multipolar potential cell to the BFUE to the CFUE. As it develops from the BFUE to CFUE, we're going to lose that CD34, which is strongly associated with blasts, as well as gaining the expression of our antigens, which you'll commonly see in blood bank. One CFUE will give rise to about 80 to 60 pronormoblasts, about just 2 to 5 days of allowing for it to be for replication. And CFUE is the last indistinguishable precursor cell. What I mean by that is any stage in this sequence here, and you see it in the bone marrow or you see it in the peripheral smear underneath light microscopy, we all just call this a blast. The first stage that we're going to be able to call it something is called the pronormoblast. So again, any precursor that is found primarily in the bone marrow or in the periphery, we're just going to call it as blasts. If you want to use more specific terminology, especially after being able to confirm what type of blast you're looking at, then you can call them erythroblasts or normal blasts. But as my previous hematology instructor likes to say, a blast is a blast is a blast is a blast. So precursors will spend about a week proliferating, maturing in the bone marrow, reticuloblastic islands until they move to the reticulocyte stage. Once they become a reticulocyte, then the cell takes about 2 to 3 days to develop and then release into the peripheral blood to live about for 120 days. So again, we're looking at these three key characteristics when we're talking about the general pattern for RBC maturation. So first is this gradual decrease in size. We're going to go from a very large cell to a very small cell. We're also going to see that nucleus go from this very nice, big, and open structure until it condenses down into about a very tight, dark blue circle. When it becomes super dark like this, it's called pycnotic. We also I tend to call it a dead nucleus because there's no way that you're going to be able to uncondense from when it becomes that condensed. We'll also notice that the cytoplasm changes from this basophilic or blue color to this pink and salmon in color. So the blue color is due to high amounts of RNA present still inside of the cell, and the hemoglobin is causing that pink and salmon color. You'll also see this pink and salmon color being referred to as acidophilic. So after the CFUE, we're going to talk about these stages, and this is what you need to know as a lab tech. We're going to go from this pronormoblast, which is the most immature stage, next to basophilic normoblast, then to polychromatophilic normoblast, to orthochromic normoblast, finally to the reticulocyte or the polychromatic erythrocyte, and last but not least is the erythrocyte, which is our end goal. So if there's one chart that I need you to memorize, it's going to be this. This kind of generally encompasses everything in just a very quick visual snapshot. Also, I just want to point out, and this might be helpful to some students, anytime they see a blast versus site in the cell name, blast means that it still has a nucleus. Site just means that it is a fully developed cell. Um, we only really see this pattern naming convention for the red cells, but hey, it might help you out a little. So the pronormoblast is by far the earliest recognizable precursor, and it is very mitotically active. It can produce up to 32 red cells over just 5 cell divisions. It is by far the largest RBC precursor cell. If I were to just, that's probably about what it looks like. Compared to its more mature relatives surrounding it, you can see I could fit like 4 or 5 in just this alone. Um, you'll also see that the NC ratio is very high. With how big that nucleus is, see how little I have for the clearing? That's also going to be a really good indicator to you that you are just looking at a very, very immature cell. Also, we call this texture in the nucleus light and lacy, where you see there's a lot of open euchromatin in there. You might be able to see these nucleoli in there. Those are also very active sites of replication. I also want to point out, and I don't really have this in the slide, but this is good for the more advanced text. You're going to see this clearing around it. That's also a very strong sign that you're looking at a pronormoblast, as well as how circular the cell is. This bottom one is a bit better at doing that you might see some weird blebbing. As well as circular nucleus. Highly circular nucleus that is very centered is usually associated with some form of erythroblast. So the cytoplasm is also very deeply blue because the amount of RNA that's inside. And again, we're having a very high NC ratio. At this point, we don't have any hemoglobin or barely any hemoglobin, and you really should not be finding this in the peripheral smear. These should only be found in the bone marrow. So now we've shrunk down a lot. So basophilic means blue normoblast. So we're looking at a blue-red cell. Well, red blast. So the NC ratio is slightly decreased, but I want to point out we have what we call these dark spots contrasted by these really light areas. That's called these are called convolutions or wrinkles. I like to think of them as almost like TV static. Also, we are still very blue because it's called a blue cell, because it's still the high amount of RNA transcripts inside. And again, you shouldn't really be finding this in the peripheral smear. This is the polychromatophilic, just means multicolored cell, so poly, multi, chromato, color, so multicolored. Normoblast. This is now the last stage that is capable of mitosis. So it's now much more condensed than its previous predecessors, and it's really irregular and very, very coarsely clumped. The NC ratio will continue to decrease as well as the actual size of the cell, and heterochromatin is now much more there. We're now, instead of looking at this just blue color, we're now looking at more this purple gray color. And that's because we're starting to increase the amount of hemoglobin that's inside of the cell. You shouldn't find these in the peripheral smear. They're typically only found in the bone marrow, but I have seen very early precursors, especially if the patient is pushing out a lot of very immature red cells at a time, usually due to medication or some sort of hormone that's causing it. Orthochromic normoblast. Ortho means correct, chromic is color. So we're looking at a cell that should be about the right color of what we anticipate a red cell to look like. So at this point, even though it has a nucleus, it is considered mitotically inactive. If you look at the nucleus, there is literally no light coming through at all. And that's because it is at this point pycnotic or entirely inactive. Also, you'll notice if you were to draw a circle around the red cell, you'll notice that the nucleus is very off center, and that's because the cell is getting ready to get rid of it, especially when the spleen plucks the nucleus right out of there. There's a very low NC ratio. You can see that we have now a lot more cytoplasm compared to the nucleus. And we're also now, again, looking at the right color because we have just about all the hemoglobin we need, only about 80%. But we're still much further along than we were in the last few stages. You might see that slightly blue as this one is, but that's just because of some of the RNA still being left behind. It's abnormal if you find them in high amounts in the peripheral smear, um, or relatively high amounts, but you can see them occasionally. Next is the reticulocyte and aka the polychromatic erythrocyte. Something that hematology loves doing is that we can't call it something unless we have the specific stain for it. So you're gonna see these big cells here with the arrows next to him. We'll call these polychromatic rather than calling them reticulocytes. But we all kind of know that we're talking about reticulocytes. We're almost to the right size here, except they're still going to be slightly larger than your red cells that's around it. And at this point, you notice that we have no nucleus, and this is considered the last immature cell in the lineage. They're still blue because they have a bit of residual RNA and mitochondria, but the remaining amount of hemoglobin will be made with that residual RNA. You can see it at very low concentrations of peripheral blood, but if you have an increased amount, then there might be some sort of condition that's causing your body to want to make up for the lack of RBCs. So this is what I'm talking about when I say that you technically can't call it something unless you have the special stain for it. And the special stain for reticulocytes is called the supravital stain. So what it does is it stains the new methylene blue that's found in the supravital stain will stain any residual mitochondria and RNA, as well as any blue-green cellular inclusions that mature RBCs don't have. We also have this biconcave disc, so it's about 7 to 8 micrometers in size. Um, or sorry, we're finally at the erythrocyte. It is shaped like a biconcave disc. This central pallor here does not mean that there's nothing in there. It just means that the center part is so thin that light will pass right through it. It's about 7 to 8 micrometers in size if you were to measure its diameter, and it has a volume of about 80 to 100 picoliters. It's also now anucleate, which means we have no nuclei, which means anything that's inside the red cell, that means that's all it has. So it's unable to synthesize any new enzymes, phospholipids, or proteins. And this is the limiting factor in the RBC lifespan, hence why it can only live for about 120 days. We're now red or pink because of the high amounts of hemoglobin, and again, that red or pink colored is called acidophilic. The clear zone inside of an RBC is referred to as the central pallor, and it should not be more than 1/3 of the diameter. If you have more central pallor than normal, then you're lacking hemoglobin for some reason, and usually it's associated with iron deficiency. If you like tables and you just want a really quick overview of each of the stages, then I would recommend this. And just sort of a quick tip for the study point. A lot of my students tend to ask me, if they're familiar with hematology. I see a lot of names for these different types of stages, and they kind of differ between what names we're supposed to go by. Well, ACP prefers the WHO naming conventions, especially for stuff like leukemias and these developmental stages. So I would recommend following anything that's labeled the WHO name. Though for students who might be familiar with it, we also talk about the FAB, which is the French and British standard. You might hear older texts saying that, but nowadays we go by the WHO's. So erythrocytes with excess iron. So usually iron will get incorporated in hemoglobin really neatly, right? But sometimes you get more iron than you really need to, and that usually can be caused by diseases, conditions, or any sort of dietary changes if you were to eat all the liver in your lifetime. So what happens is that any excess iron will start depositing on the RBC membranes, and they might start depositing on not just your red cells, but also on your organs and other tissues of the body. When we notice that these iron depositions start building up, we'll start to see them on the RBCs as well. So cells that have an iron deposit is just called a sidero something. Cyte means cell, so that's usually for, in this context, talks about RBCs without a nucleus. Blast means that we're talking about RBCs with a nucleus. So siderocytes just mean any iron deposits found on RBCs that don't have a nucleus. And sideroblasts just means that we're looking at iron deposits that are found on any RBCs that still have a nucleus. The iron can only be stained and visualized using Pearl's Prussian blue, and normally your spleen will do a good job at removing these cells via culling and pitting. So this is what the siderocytes look like. So down below is the siderocytes, and you can see that the cells don't have any nucleus, so I'll call them siderocytes. Meanwhile, these guys with these big round nucleuses, those are sideroblasts because we're talking about the iron deposits that are found on them. So the erythrocyte membrane is made up of this phospholipid bilayer. So when we talk about it being ampiopathic, that means it has two ends or parts. So the hydrophilic polar heads will face the outside of the cytosol or the inside, while the tails will face each other, sandwiched in between the membranes. The polar heads are hydrophilic, and the fatty tails are hydrophobic. And the reason that we talk a lot about the membrane is because it's really essential for proper development and function. RBC precursors require EPO, cytokines, growth factors, and transferins in order for it to grow and develop. They also require ions, enzymes, and substrates in order for it to live, and RBCs also need to be able to expel waste properly. And all of these are reliant on having a good RBC membrane. So one of the biggest functions is that it has to provide the right amount of structural support and flexibility, so that way the red cell, as it's going through these really small and tiny capillaries, it'll still maintain its shape without deforming too much. So it's made up about majority of proteins, next is lipids, next is carbohydrates. And any sort of defects in this composition, especially if you have more lipids than needed, then that might cause compromise in in the integrity of the membrane, and that might end up in premature cellular death. So there's a few things that we talk about. In the phospholipid bilayer, especially in your red cells, we notice these things. So in the outer layer, we have sphingomyelin and PC, and in the inner layer, we have PE and PS. Cholesterol will be sandwiched in between the phospholipid bilayer and will be present on both sides. The amount of cholesterol will affect the cell fluidity and permeability. So think of this like making a cake batter. If I have too much batter, then I'm gonna just have too much cake. So what happens is that you get so in codocytes, right? You have too much lipids overall. So that's gonna cause this membrane to want to fold up on itself in a really weird way. So what it looks like from the surface is that you'll have this target cell like effect where this is just the excess amount of membrane that's folding upwards. Codocytes, by the way, thanks to my students, codo meaning bell, and the reason that it calls that is from the side it's supposed to look like a bell. I don't know how much you're supposed to see it, but hey, that's what they're called. If you have excessive amount of cholesterol with no increase in phospholipids, then that will cause acanthocytes, which again, they're not very fun looking. Acanthocytes meaning thorny, so you're going to get these thorn-like projections from it. But again, think of these like cake batter. I don't want too much and I don't want too little. If I have too many ingredients, then that's going to cause my batter to end up acting a little weird. We also want to talk about the relationship between the outer phospholipids and the inner phospholipids. If you have too much outer phospholipids, then you're going to end up with this weird membrane um, where it looks like burr seeds, if you ever had to deal with those. Those are called echidnocytes. They're called that way because they look like the animal, echidnas. Excuse me. Stomatocytes are also what happens when the inner membrane phospholipids increase instead. Stomata meaning lips, so these just look like big old lips. Glycolipids and glycoproteins act like your antigens, so they play a huge role in cell signaling, activation, and cell recognition. And you'll talk a lot about these, especially in your blood bank. But these are what we call your antigens, and this is what blood banking is all about. Fortunately, we're not a blood bank class, so we won't really talk about too much. So the membrane has two different types of proteins. We have these integral proteins that traverse the entire membrane versus the peripheral proteins, which are only attached to one side of the cell membrane. Most proteins are only found on the cytosol side, and their job in, in to be attached to integral proteins. And again, RBCs can only synthesize all the proteins that it ever needs right before it loses that nucleus. Once that nucleus becomes pyknotic and is extruded, then you literally can't turn back. So peripheral proteins are found mostly on the outside or the cytosol side of the bilayer membrane. And a lot of these serve either enzymatic or structural roles. So structural proteins are organized into this lattice formation to form the skeleton. And just like the human body skeletons, its job is to give it the strength and support to the membrane that it needs in order for it to maintain its shape. It'll also play a major role in its ability to twist and turn, and that's called deformability, as well as stability. Any defects in the cytoskeleton are usually associated with some sort of weird shape, decreased stability, and hemolytic anemia overall. So this is just a very quick, brief look at each of these proteins that we commonly talk about in heme. If you want a more in-depth look, you can always look these up, but this is about what I usually expect students to know. Band 3 is a anion exchange proteins that exchanges intracellular and extracellular anions, and its job is to transport bicarb. Spectrin is the predominant cytoskeleton of the RBC, and its job is to expand and contract the membrane. because it acts like a spring, spectrin spring. Integrin acts like an anchor, and its job is to stabilize all the proteins that I just talked about. So if you're into biomolecular, you can look at this if you want a more in-depth look, but for more lab techs, you shouldn't have to worry about this too much. So all the components that I was just talking about, they have to work together in order to maintain the RBC's integrity and deformability. It has to be strong enough to withstand any turbulent circulation within the blood cells. So what the RBC needs to do when it goes through these really strong circulation and these really tight capillaries is that it has to be able to maintain its shape. So the ability to twist and squeeze and then pop back is called deformability. And deformability comes from these three traits. First is the biconcave disc. Next is the viscosity of its internal contents. And last but not least is the elastic properties of the RBC membrane. Coiled spectrum will allow for elastic extension and allows the cell to resume its shape. The RBC membrane is freely permeable to water and anions, while the RBC membrane is impermeable to any cations. But in order to maintain the right amount of concentration in its cells, the RBC membrane has to use these cation pumps. So its job is to keep sodium and calcium low inside of the cell, but keep potassium high inside. Since more positive ions are leaving, then the cell actually gains a slightly negative charge, and that way it just helps prevent ions from just leaking out of it. But this is sort of what we talk about. So when your cation pumps stop working, it can happen due to ATPase failing, the cation pump actually failing, or just not having enough ATP. When that happens, then water will start to just move in and out the way that it likes. And the easiest way I like to simplify this is that water likes to follow stuff. So a hypotonic solution means that you have less stuff on the outside of the cell than you do the inside of the cell. So when that happens, water will want to rush from the outside in, and that causes these cells to burst. In a hypertonic solution, you have more stuff outside of the cell than you do the inside. And what will happen is that water will want to leave the cell, and that'll cause these cells to shrivel up or crenate. And if you're in the just right solution, then water won't want to move one way or the other. So we're going to talk a lot about the shunts that are associated with the RBC's health. So the main function of red cells is that it has to be able to bind and carry oxygen from the lungs to the tissues and remove the waste from the tissues and allow organs to excrete it. But the thing is that these processes are not passive and do require energy. So how do RBCs get energy in order to be able to survive when they have no organelles, mitochondria, or nuclei? So these are the four pathways that help the RBC to, well, basically exist. And we're going to talk about all of these right now. So this is a really quick snapshot. If you have to memorize one thing, then overall about each of the four shunts, then I would suggest looking at this slide. Of course, you're going to need to know a bit more detail about this, but if somebody were to tell you, give me the quick rundown, then this is a perfect slide. So the Ebi-Meinhof or the glycolysis pathway basically just allows ATP to be generated from glycolysis, and that passively produces lactic acid. The hexose monophosphate shunt keeps hemoglobin safe from oxygen radicals. The methemoglobin reductase pathways, its job is to keep iris in the ferrous state, which allows it to bind to oxygen. And the Rappaport-Leuenberg shunt, its job is to keep hemoglobin stable by keeping it in either its tense or deoxygenated state. All of these pathways together, you can't lose one and just expect the red cell to survive. All these work together to help maintain cation pumps and the electrical chemical gradient keeps the hemoglobin stable in its ability to bind to oxygen and maintains the RBC membrane integrity and deformability. So I'm just going to call this the glycolytic pathway, but understand when you see Eben-Meyerhof pathway, you should be thinking glycolysis. This is the primary way for RBCs to maintain ATP from glucose. RBCs don't have any glycogen storage in order for it to store up glucose, so it just entirely depends on free blood glucose. And 95% of that RBC's glucose that's found inside of the cell that it's able to take up will be converted to make ATP. So what happens is we convert glucose into 4-ATP and pyruvate, which as a byproduct also produces lactic acid. We only gain a net 2 because we have to have 2 in order to start, but hey, net gain is better than zero gain. But ATP again is, if you might have remembered from your high school biology class, is the money for the cell. You need to spend it in order to maintain RBC shape, flexibility, and integrity. The hexose monophosphate shunt, its job is to detoxify any peroxidide groups to help prevent any oxidative damage in the RBC. And the biggest thing that we talk about in this shunt is the G6PD, which is an enzyme found in the HMP shunt. And basically what it does is just helps reduce NADP into NADPH. And the way that it does this is performing redox reactions with, with hemoglobin and the reduced and oxidized form of glutathione. So what happens is if you have a defunctive HMP shunt is that it will cause hemoglobin damage. And that will cause the hemoglobin to denature and land right onto the RBC membrane. So what happens is that this is not meant to be there, right? And your body knows this. So when that protein binds to the membrane, that will cause the membrane to start becoming leaky. And the cell actually loses ATP trying to maintain the cation gradient. So what happens is that these denatured hemoglobin bodies or Heinz bodies attaches to the membrane, and their splenic macrophages are good at recognizing what's meant to be there and what's not. So they'll actually just take a chunk where the Heinz body is and form these bite cells that are found on the top peripheral smear there. Methemoglobin reductase, its job is to maintain iron in its reduced form. So when we talk about reduced iron, we call it Fe2, which is Fe2 pos. Fe3 is the oxidized iron, and that's called Fe-ic. And the reason that this is important is because methemoglobin cannot bind to oxygen. Only ferrous can. So what it does is that it helps keep the iron in its reduced form, because if we have Fe-ic, like ew, then it's not a good thing. And the primary enzyme there is methemoglobin reductase, which reacts with NADH. So what happens is that if your methemoglobin reductase pathway isn't able to make up for the amount of methemoglobin that's building up in your body, then your blood won't be able to carry as much oxygen as it normally does. And that causes signs and symptoms starting to manifest. So what happens is that your skin will start to blue, and that's called cyanosis. And this is strongly attributed to anything that has decreased oxygenation in your tissues. Rappaport-Lewinberg shunt. Its job is to produce 2,3-DPG by converting 1,3-DPG. Well, what do we need this for? Let's say that we take a hemoglobin here. Normally it has a space for oxygen to want to bind and go into. 2,3-DPG, however, gets in between the spaces in the hemoglobin, and it causes it to become tense, just like this. And by binding to the inside like that, it'll affect the hemoglobin's affinity to be able to bind to oxygen. So when 2,3-BG binds to hemoglobin, then that'll cause oxygen to be released. So therefore, the hemoglobin affinity to oxygen will decrease. But when it doesn't bind to oxygen, or when DZBG doesn't bind to hemoglobin, then it gives that space for oxygen to be able to bind, and therefore hemoglobin affinity to oxygen will then increase. And it plays a vital role in oxygen delivery to the tissue to ensure that hemoglobin will actually give up its oxygen. If you didn't have 2,3-DBG, then hemoglobin's more than happy to carry around that oxygen forever. So this is what actually gets the oxygen to come out of the hemoglobin. So if you just want a very quick table, then this is a really nice table. It'll outline the name as well as its key enzymes and its function, and if something were to happen to it, then what sort of pathology will it cause. I'm not gonna make you memorize this. I believe the ACP doesn't really expect you to memorize this either. Just understand that your red cell will not be able to exist without any one of these shunts. If one of these goes out of whack, then your RBC won't be able to survive for as long as it needs to. So this is where I talk about a bit about kinetics and concentration. So this is represented by the oxygen dissociation curve, and it's just a visual representation between hemoglobin oxygen affinity and concentration of oxygen in the peripheral blood. When you have a left shift in the dissociation curve, then hemoglobin's affinity to oxygen will then increase. And this is usually associated with any sort of condition where your body doesn't need as much oxygen. And this will happen when temperature will be decreased, 2,3-BT concentration has gone down, and you have an increased pH. In a right shift, hemoglobin's affinity to oxygen will decrease. And this is associated when your body needs oxygen more. So this will occur when temperature is increased, concentration of 2,3-DBT is increased, as well as a decreased pH. So, sort of a question I like to ask my students is what direction do we expect the curve to move in when you're exercising? Pause now if you want to figure out for yourself. All right, if you answered right shift, then you would be correct. So, in exercise, your temperature is going up because you're generating a lot of body heat, and your body's releasing a lot of lactic acid, which will decrease the amount of pH, which will decrease your pH in your body. And this is what the oxygen dissociation curve looks like at a very broad level. So understand that when we talk about what's normal, that will vary largely by your demographics. So it'll mostly vary because of your age, your sex, and geographic location. So what happens is that when you're born, your RBC and hemoglobin will be much higher, and your reticulocyte can also be much higher than you normally would have as an adult. As you start to age, we have this like physiological anemia in the newborn. That just means that your red count will start to decrease until it resembles adult reference ranges. Male patients will also usually have a higher RBC count and hemoglobin concentrations because testosterone resembles a bit more like EPO to your body than estrogen does. It's not due to any other physiological reactions. If anybody says so, then tell them no. Regulation and production. So we need to make sure that we have a right amount of cells. Not only do we need to make sure that they're functioning, but we have a right amount of cells. So you can do this by either manipulating the rate of the production or manipulating the rate of release. Any sort of increased rate associated with either or might be due to sort of decreased oxygen saturation. So this will usually happen when for a variety of reasons. Generally, your body just needs more oxygen than it currently has. So this will occur in any conditions like anemia, heart and lung disorders, hemoglobinopathies, high altitudes, and acute blood loss. The primary hormone that'll regulate this is EPO, which is secreted by the kidneys when it detects that your body is undergoing hypoxia. And during any time that your hemoglobin is less than 12, then EPO will also increase very sharply. Decreased rates of production are usually associated with some sort of bone marrow destruction, renal disease or failure, or aplastic anemia. So there's sort of four main functions to EPO. Its job is to increase the amount of BFU to CFU mitosis and maturation, increasing the speed at which mRNA is being translated into hemoglobin, decreasing the maturation time, and finally stimulating the early release. And sort of as a fun byproduct, if you have chronic renal failure, then you might also experience anemia secondary to the renal disease. But generally, if you have a high amount of EPO, then that's going to cause these four conditions to happen all together. And that's why when you see patients have a high amount of EPO, then you're going to see a lot more immature cells in the periphery than you normally would in a normal body. Erythroid destruction, and this should be the last section for this lecture. So, destruction is good when we're just getting rid of non-functioning cells and aged cells. Usually this will happen when the RBC is sort of approaching its end of life, and that the four shunts over time have just stopped working as effectively as it needs to. The thing is, though, that when RBCs are about to be dead, they need to be broken down systematically. Um, Sort of for two reasons. One is so that we can reuse the stuff that we used to build it back up. For example, we could take apart hemoglobin into heme and globins. Heme we can break down into its iron and bilirubin, and we can reuse that iron. And globins are converted back into amino acids, which we can use later on. Also, we need to prevent damage to the cell. Hemoglobin is not meant to be outside of a red cell, and it'll cause a lot of problems, especially if it's left alone in the peripheral blood. So, this is the way that we want to get rid of cells: extravascular hemolysis. This is a normal process, which will happen primarily in the spleen. When we do extravascular hemolysis, we can also return the RBC components back into the bone marrow, so that way we can continue making new cells. And majority of your cells will go through extravascular hemolysis. So what happens is that your RBCs go through your splenic red cords. And the splenic red cords have this hypoxic and acidic environment. While good RBCs will be able to cope with a little bit of stress from the environment, but older RBCs will not be able to. And that allows the macrophages to go in and scoop them up and colon pit them. And again, we prefer extravascular hemolysis. That way we're not just having things bouncing around in your body to do whatnot, and we can also reuse the components that make up the red cell. However, not everything can happen the way that we want to. So intravascular hemolysis is when the RBC gets destroyed inside of the red blood vessels. And when this happens is that it might cause inflammatory responses, or DIC. So severe trauma to the membrane, structural defects, toxins, drugs, complements, or antibodies can cause the RBCs to become spontaneously lysed inside of the red cells. But again, we don't really want red cell destruction to happen this way. So what happens is that the RBC just pops open like a balloon, and it just starts spilling the inside of the red cell into the bloodstream. And the reason that we don't want this is because the hemoglobin can become bilirubin. And if you've seen bilirubin crystals, they're very sharp and really not meant to be anywhere outside of your liver. So when that happens, it'll deposit on tissues and organs, which will cause damage. And if it's really bad enough, then it can cause organ failure. But your body's good at coping and surviving. So your body can compensate and clean up intravascular hemolysis by using acute phase reactants. So the biggest ones that we talk about are haptoglobin and hemopexin, whose job is to transport bilirubin into the liver. When bilirubin is transported into the liver, then we can conjugate that bilirubin and excrete it into the biliary duct or filter it out in our urine. And these are the labs that you want to be ordering if you're trying to figure out if this patient is having an increased amount of extravascular hemolysis or intravascular hemolysis.